Hello everyone, this is Cupcake J. Today we're going to talk about Unit 3 of AP Biology, Cellular Energetics. Enzymes are complex proteins with a specific shape that must have the correct substrate in order to decrease activation energy. Without a biological catalyst, the reaction rate would slow down as more energy is needed. If our enzyme goes out of its optimum range of pH and temperature, denaturation will occur and cause the enzyme to change its shape. Therefore, the substrate cannot fit. A competitive inhibitor will bind to the active site and therefore block the original substrate in binding to the enzyme. A non-competitive inhibitor will bind to an allosteric site outside of the active site, causing the enzyme to change shape and therefore the substrate cannot bind to the enzyme. Photosynthesis is a process used by plants to make their own food. Plants take in raw materials of sunlight, carbon dioxide, and water to produce oxygen and glucose, a 6-carbon sugar that provides short-term energy used to carry out other cellular functions. Here's the chemical equation of the process. 6CO2 plus 6H2O yields glucose and 6 oxygen molecules. As a whole, photosynthesis occurs in a plant cell's chloroplast. However, photosynthesis can be broken down into light-dependent reactions and light-independent reactions, or the Calvin cycle. As stated before, photosynthesis results in the production of glucose and oxygen. These two products are used by mitochondria for cellular respiration, which is a vital process for living things. Overall, it can be said that light gives life. Cellular respiration is the process used by all living organisms to use the energy stored in biological macromolecules to produce ATP. This process occurs in the mitochondria and involves glycolysis, the Krebs cycle, and the electron transport chain. Here's the equation. Glucose plus 6 oxygen molecules yields ATP, 6 H2Os, 6 carbon molecules, and heat. Cellular respiration starts in the cytoplasm with glycolysis, which breaks down glucose into 2 pyruvate molecules, 2AP and 2NADH. In case there is no oxygen available, fermentation will occur for glycolysis to continue. Alcoholic fermentation is used by yeast and produces ethanol, while lactic acid fermentation makes lactic acid. This pyruvate is oxidized into acetyl-CoA and then moves to the mitochondrial matrix for the Krebs cycle, where it is oxidized again, resulting in CO2, ATP, NADH, and FADH2. The electrons are finally accepted by oxygen at the end of the ETC, and water is formed. By chemiosmosis, the protons move through ATP synthase and are combined with ADP through oxidative phosphorylation to make usable ATP. Yippee! Energy! The laws of thermodynamics say energy cannot be created nor destroyed, and that entropy increases in a closed environment. Autotrophs get a constant supply of free energy from the sun, and heterotrophs acquire this energy by eating said autotrophs. All in all, homeostasis is maintained through a constant supply of free energy to bring proper order to the world.